Hey folks, how are you all? So I hope you are all doing well and all your work is doing awesome. It's been quite a while now since we have been doing this on AI for bioinformatics, AI for research. Actually, we actually want to cover a lot and lot of new techniques in order to get this, how to see this kind of an evolution that we saw in our entire workflow of this hustle that we are supposed to be putting in order to get the drug development and all these techniques evolve. So every day it's a whole new level of challenge. Every day it's a whole new level of task because drug development takes years and years of evolvement. Actually, it's like humans who are involved in quite a while now have actually never been that successful in getting a drug which is doesn't have any side effects. So independent researchers need not have to worry, so their point of view differs. So let's just start with the session. Let me just share my screen, and there's a whole new level of complexity that is involved every time when you're working on something called as a problem statement. So problem statements come out in different forms, so it need not have to be something which is more simple, it need not have to be something which is complex. So in this session, let me get your what is that, your dreams right. Here in this session I can't completely explain you about what is going to happen or what is not going to happen in drug development. So of course I'll be actually doing an in-person session if possible, if the pandemic goes away and we have a lot and lot of things coming up. So yeah, that's what I really wanted to do. So let me make sure that I'm sharing my screen properly. So application window, yes, I'm sharing my screen. I'll add it to stream. Yep, we are live now. So let's just go. So when it's all about the drug development, it's all that approximations that come into the picture. It's all the biological approximations that you see in your everyday life, because when you are thinking of something called as a solution, you are supposed to derive everything that is very close to an human approximation. It need not have to be something like, okay, I'm just delivering this drug to a person. It just needs to work. There's nothing called as a miracle or a magic ingredient that you can think about in your everyday life. It can be very complex because every day when you're thinking of something called as an application which you can really think of, from an AI to the field which is nowhere related to AI, I think it's very complex to design such application. So when we think of such applications, we need to know what are our problem statements that we are going through. We should be clear with the techniques that we are supposed to be giving in, and we also should be very sturdy enough that our decisions are not being affected by anybody. Because when you think about making decisions in the real time, there can be anything and everything that can go wrong, because every time when a new molecules gets released into the market, it's like 15 years of prior research or else 5 years of minimum prior research that goes in. There's no prodigy in this field because yes, it's very complex. So of course there's no prodigy. That doesn't mean that scientists like me who is an ECE student can't work in this field. it's like it's like you depriving yourself from your right to work in this field. So I think probably I should be knowing which stream are you guys from. So in the chat box, if you can just share me which stream you are from, I think I can channelize my thoughts there. So if you are shy, don't worry, you can ping me in LinkedIn if possible. So my LinkedIn profile, I'll be posting it in the comment here. So yes, I've posted my LinkedIn comment here, so you guys can see it. So you can post me in the LinkedIn, so you can streamline my talk for what I really need to focus on. So let me get the banner on. So Pi and AI Bangalore chapter will be hosting a lot of such events like this. So make sure that you follow our LinkedIn page and also subscribe to our newsletters from DeepLearning.AI. So yeah, that's what it's. So let's keep rolling, guys. So influence of drug delivery and the development, and also biotechnology in this field of pharmaceuticals. What will be the future potential that we will have to look at, and what will it look like? Patient centric drug discovery and development. So yes, this is the overview of what this introduction to this technology is all about. Of course, we can't be doing everything in 1 hour or 1 day, because it took me a lot of time to understand one problem statement which was there in Kaggle. It took me a very long time to understand what is there. So let me just break down this every little point that I've put in, so that you guys can get to know what is that actual thing that is actually I'm trying to convey here. So influence of drug discovery and the development is basically one such technique where it's been there from quite a while now. Let it be from the ancient ages where anesthesia was being discovered, or else about something very close to like penicillin which was discovered. So Louis Pasteur is one of the person who gave vaccine for your rabies. So pasteurization is one of the techniques where it is used for delivering your everyday milk. So it's like there can be virus which can be prone to live in some extreme situations, not that extreme in such a way that it can withstand high temperature. Some virus are there where it can never withstand lower temperature. So keeping all that in mind, pasteurization was done. It's just simple technique, boiling it to an highest boiling point and freezing it back. So that is where you are actually getting the pasteurized milk and everything coming up. So this is where influence comes into the picture. It need not have to be complex, it just can be that common sense that you're thinking about in order to deliver something. It need not have to be so complex, so sophisticated in terms of research, filled with machineries. Of course, when you are doing drug development, it's a whole new level of complexity involved there, because every drug which you are doing as an research, where we are trying to create a new drug or a new molecule, it needs to be done in probably class whatever, probably 10,000. It's like should be the cleanest room that you should be. So yeah, I can share one thing. Like, I've done a podcast with a person called Dr. Rajesh Lal Chandani. Of course, you can connect with him if possible. So he has given that amazing podcast. Of course, he's a dermatologist. That doesn't mean that he should never be doing anything as such. So he is pretty good in his medicine. He works on botulism toxin. So you can check this podcast link which I've given here, so that you guys can get to know what's that pretty big chance of you as an engineer not overlaying yourself with too much of responsibilities, going back to the world of medicine. Of course, you need not have to have a degree of biotechnology or anything. You just need to just have that mindset of working. So influence of biotechnology in the field of pharmaceuticals. Of course, we have seen how robots have been used in delivering drug. We have seen how origami has influenced robots to give an amazing influence in terms of surgery, because origami is one technique where you can fold an paper in any form and get it in any form at your desire. So origami has made robotics evolve. Not just origami, actuators have evolved these days, so you need not have to worry about anything and everything that you are thinking about, whether this actuator will work for me or not, whether this robot will be able to deliver me the right thing or not. It's not that. It's all about your actual work which you're thinking. So at the end, I think probably I'll be going to the insights of the data sets. I think probably that might help you all in order to get that. And what will be the future potential that we will look like? Future potential of this technology involved is like you are being reduced with the complexities. You are not overwhelmed by this AI in drug development. Don't get overwhelmed by this technology, because this is not going to be anything that is as influential as a person coming out with a new drug. It's not going to be that, but it's going to ease the work. It's like probably people took years of experiments to analyze a disease or something, like good amount of people putting in their hours and hours of work. It's not like that. You just need to just go there, start to look at, okay, this is supposed to be what I'm working, so yeah, I need to get these things done. So that is how technology is evolving. So make sure that you have a good mindset in your actual work that you're going to deliver tomorrow or any day in your profession. So you're not falling in love with the technology, you're actually working in order to deliver a new technology to the world. So patient centric drug discovery and development is one of the thing which actually came up, because today when you see progeria is actually a not a disease, it's actually a genetic variation that an person goes throughout his life. So when you think of something like that, it's all about that work which you are giving in. So when you think about something like technology being influential, of course yes, it's going to be influential, but how long? Because precision medicine is in a picture, so it need not have to be that complex in order for you to deliver something new. It's just going to be a simple magic that you're going to perform. So anytime, anywhere that you're thinking of something like, I need to do some groundbreaking research, you need to make sure that you're looking at a field which is like more unique. Don't follow the crowd. So now when you think of patient centered drug discovery and development, there are lot and lot of genetic diseases which people have discovered. Genetic ailments, actually they are not diseases, they are just ailments where there's a mutation of one gene and it starts developing throughout the body. So cancer is one such thing. So people might be suffering from different types of cancer. So it's all in you whether you want to choose this or that. So choose the right one, right problem statement, and start working. So now when we look at all these, investigation of the new drug application is one such technique where research is being involved extensively. It's not about whether you're 100 years experience in pharmaceuticals or 100 years experience in terms of data science. It's all about your actual will to understand the subject domain and start working. So investigation of new drug application is an whole new level of research, because drugs are supposed to be designed for something which is more specific, but these days it's all about one drug, many problems solved. So probably there is going to be a lot of side effects in that also, but we have to design by using some sense called as AI. It's the new sense. It need not have to be like people making decisions every time. It's like you throwing in some data and the model coming out with the insights of it. So pharmaceutical and life science strategic plans is basically one such technique where you're thinking about where can I get the feasible application of this entire portfolio of, what can I say, this problem statement. Whether should I use computer vision here, should I use a microscopic image, apply the computer vision techniques, get the insights of the data set, or should I use some sensors like EMG, EEG, EKG, or anything which is there. So if you want to talk more about my sensors and all these, I think probably my LinkedIn has lot and lot of featured workshop materials which I have. I think probably that is going to help you all. Just go to the featured section, just go to all. I think I have kept all the slides here, so you can just go there and start working on these things, so that you will have a clear cut thought of, okay, I should be working on these problem statement, I should be working on this, I should be working on this part of the problem statement only. Because I'll be very frank with you, it's a whole new level of complexity. I'll be showing it in the future slide, so wait for it. So folks, so let me just go for the next slide. IND, independent uh, applications, and all this new drug application is where the development comes into the picture, where you're not supposed to justify everything just because you're doing, like, you just need to go there and you just need to start working on every clinical study and the design. and you need to have a clinical approval in order to get the research. I'm an open source researcher. Trust me, I don't have an access to the clinic, but I have an access to the data set. Cancer Imaging Archive is there. There are a lot and lot of Physionet is available. There is lot and lot of scientists who have open sourced their data sets. So you can just go there, work on those data sets, get those insights. So this is where your clinical data study comes into the picture. So now when you think of something as an application, this is the complexity where I can say every time when you think of this, first is basic exploratory. It's all about the target. It's all about the lead ID. It's lead IDs are basically one of those research entities where it's going to be very helpful when you're going to go for your patent, where you're going to go for your insights about the new drug which can be developed using your present developed insights from the data set which you have got. So it's going to be helpful in order to get that thing done. So Kaggle has got lot and lot of data sets. You can start working on it. Mechanism of action is one of the data set which I'll be doing. So in this, I'll not be going too much detailed about it. I'll be doing data analytics part of it. It's all about data visualization. Sorry for it. It's like how much is the insight that I can go. I've only used one thing called as principal component analysis. I think it's not, I hope that you guys have a basic knowledge about machine learning. So I'm skipping that. So I'll give you an entire Google link. Just go there, just start working on it. So this will be the entire Google link. So I'll be pasting it here. So this is the one, folks. You guys need to copy it. Just go paste it on your browser, or let's just go type it as principal component analysis in Google, and Google is going to come out with lot and lot of insights. So I'll do the work easy for you. So just go paste this thing. Everything is going to come to your way. And now when you go for the future, there is something called as phase 1 of clinical test, where you are going to do an human research. Basically, you're going to do it on a white rat or a guinea pig. So this is where your research comes into the picture. So phase 1 clinical tests are something which is there. So phase 2 is just like the cross validation technique, where you have train, test, and validation. Just like how you're going for a patent or in publishing the paper, you work on your validation data like 1 week or 2 week prior to the submission, right? So this is where phase 2 comes into the picture, where you're actually working on the actual insights that you have got, and what are your desired insights, and are these results matching, because you are supposed to come with matching results which are going to be much more helpful for you in the future, right? So this is how you're supposed to be working. So phase 3 clinical tests, all these things, what might be the span of time that it takes for any researchers to come. So all the participants, I think there are 22 participants here. So anybody who can give some insights here, like anybody who can give some insights, like let me not waste time. It takes years and years of research in order to give you this insights. So every time when you're going for a clinical trial, you need to have a subject domain expert. That's a doctor or a pharmacologist who is working on a drug which is like very similar to what you are trying to discover. So AI and drug discovery, yep, it's going to be an humongous work, because every time when you're thinking of such kind of an application, you need to have a subject domain expert near you. So that is how it's going to help you. Let me just go to the next slide, which is your problem statement. Your problem statements are in plenty, my friends. There is a huge deal in terms of problem statement. You need to have right subject domain knowledge. You need to have basic need to understand the problem statement. You need to have a right understanding of what the frameworks that you are using. It can be TensorFlow, Chainer, Kefi, Theanos. Of course, Theanos, all these things are not being used, but if you are using it, you need to know the insights of what is going to work and what is not going to work. If you are not going to do that, nothing is going to work, because right framework is like 50% of your work. You know which framework to use. Of course, I've used sklearn here because sklearn has all the optimized code and everything done. So it's just like you just consolidating the data as X test, Y test, X train and Y train, everything just throw in, you will get the insight. So make sure that you have this slide bookmark in your, I'll give you the picture. I think probably I can share you the slides. Yep, I'll share you the slides in the description below so you guys can get to know the actual workflow which is going to be there in this technology. So let's just move on to the next thing. Next thing is all about the problem statement. It's, you need to work smart, you need to put in the work of understanding the above factors which I've told in the previous slide, and you need to be very hardworking. You need to be very hardworking, folks. It's like, in this field you can't have approximation make a huge deal here, because it's like Bill Schmarzo saying, I was there in Power BI working with absolutes, and he goes to Yahoo, and it's a whole new level of approach. So yeah, it's a different thing. So yep, let's forget this. Let's go a little hands on. So I'll share my Chrome tab. Where are you, Google Colab? I think, whoa, it's open, right? So AI in drug development, yeah, everything is here. So up, sorry, it's named as data visualization. Sorry for that, folks. I'll reconnect it. So in the meanwhile, I would love to ask you, like, which branch or you folks are from? Yeah, there are 24 participants here. Like, which branch are you go folks from? Yeah, if you want, you can answer us. No worries. So I think it's reconnected. I think the drive was not mounted. Yeah, drive was not mounted. Yeah, it's like I didn't connect it, right? So I have lot and lot of things. So I'll mount the drive and I'll come back to you. So I take the code and I put it here. So done. So I've mounted the drive. It's going to take a lot of time. So mounted, yep. And yes, I'm going to unzip that, because yes, it's unzipped, and I'm going to show you how to mount it also. It's very easy, folks. Let me just say, it's all these Google, all these GC, Time, Pandas, all these frameworks I'll be using in next one, which is data analytics. I'll be not using this because I'll be doing a simple data visualization using, and I'll be applying a simple formula called as PCA. So principal component analysis is what I'll be using. So you guys can have a brief intro on what this technology is all about and everything. Why is my screen so less here? Okay, fine. I think probably the problems of sharing that application. Yep, I think so. Yep, it's a problem of sharing an application. So let me just go back here. So I've taken train feature, train target score. So all these are the ways in which I'll be just reading the pandas with the CSV file. So pd.read CSV is just going to give you that function. It's going to read that CSV and all these, right? So import CSV also is there. So let me just give you that quick intro of MOA, Kaggle, mechanism of action prediction. So I'll stop sharing that screen and I'll come back here and I'll share this screen here. It's Kaggle MOA, mechanism of action. I think I'm sharing that. Yep. This actual thing is very unique challenge in Kaggle. I think I've come across a lot of challenges. I think this is the one of the unique challenges that I've seen in this entire work. Because NIH has one of the institution which has given out all the leverage that you can really think of when it comes to research in drug development, stem cell, regenerative medicine, and all. So in the past, there has been a lot of problem statement which the people have seen. There's quite a different problem statement where which determines the mechanism of action. Mechanism of action is basically where you're just trying to predict what might be this entire behavior of a molecule that you are going to synthesize. So Let's just go for the train features. These are CP values, CP doses, significant IDs, and all these comes out in a real time application. When you are looking for this, it's an ID and TRT-CP. If you want to know what is CP in drug, it's like carbon family and all these things. So drug discovery is basically a new dimension of where you're thinking of about that research. It's like the ID for your drug bank. So this is where they come into the picture, and all these are some of the features, and we are thinking about. So these are the time in which the CP doses have been there, and these are the dose one. D1 is like dose one, D2 is like dose two. So how many D1s is like 51%, D2 is like 40%. Of course there is some skewness in this data of like 2%, which is like above here, below there. So you need to target it. So this is where I come out with all these kind of techniques of making sure that I get the right insights for all these development that we are doing. So yeah, when we are training the non-scored entities, we come up with all these inhibitors, transporters, and all these techniques combined together will give you a lot. So all these zeros are like there are no side effects, and all these targets are completely fine. These are supposed to go for a human trial, and all these insights will be there. So test features is one where you will be going for the same CP, same dose, but you will be testing some data. So these are like sure shot predictions that are there. These are probably like 99.99% accurate values, completely like that, or else 90% accurate values. Who knows? Anything can go wrong. So this is where it comes. So let me just share my code screen. Oh no, I'm not supposed to invite any guests here. Sorry for that. I'm supposed to share my screen. So Chrome tab, data visualization, share. So of course, when we do that, we are supposed to optimize it. So when you're supposed to optimize the code, you're supposed to do it, because pandas is not horizontally scalable, just like your Hadoop and all these. So you need to streamline your memory so that you don't get too much of overhead for your RAM. Of course, there is active session which is going on. There is 0.91 GBs of RAM which is being used. Fine, absolutely fine, because it's where you start working. So let's just go on. So train features, and oh, sorry for that, I didn't run the previous ones. Oh, I forgot to run this, right? Yep. So it's now starts working. So train features, reduce memory usage, and all these will come. So memory usage decrease to 5 and all these. So you should be knowing all these basic terminologies in your this one. So this is an incomplete notebook which I have built. Sorry for that. I could not comment much. I could not do that because my exams got finished on 27th of Feb. Yep, 27th of Feb. And I was filled with lot of other work, so I could not do it. So yeah. So let me just start running this. So when you think of all these, there's display of features, all these coming up. So the quantity of missing data are one. So you can also use pandas function as .nan or .none or anything. It will start working. So we have just gone the rough side of development. So yeah, display, test, and everything comes into the picture. So test value is also done. So in order to get yourself that one implication of, okay, you're working on the right set of approaches, so we are supposed to segregate the data, we are supposed to reset the indexes. So it's like common for every problem statement. You need not have to worry about, okay, this is not working, that is not working. Your variables can be same. There are hundreds of people who can write the same code in the same style, but like optimizing it in a different fashion, yes, it takes a lot of time. So when you think of these bar charts are going to give you a lot of insights on patches. So patches in your cloud are different. Patching is like updating and all these, patches are like you're actually getting that every insight of the data. So the maximum value is 17500 and it's 90%, and it's TRTCP. It's the ID which is going to be there, the CP values. The CT vehicle is not there, so it's like one such entity where you will be thinking of, okay, should I give the doses or not? So that's where it comes. So for more information, just go back to this entire notebook of data which is there. Lish MOA. It's actually MOA. So they named it as Lish MOA. So, oh, Ritesh Yadav is here. So, hi Ritesh, how are you, brother? Fine. MOA is an awesome competition in Kaggle. Thank you so much. And I work on Google Colab mostly because I love Colab. It's like it's like give an extension, you don't even open the tab, it starts working and it will give you some results. So that's where I use Colab for most of my submissions. Yeah, when you take features, take without control, and it's basically visualization that you're thinking of when it comes to that. So validation of features without the control is validation of the feature set, drop, and all these you're just copying all these in terms of this variable. So this is where it comes. So without control, yep, you're going to do that. And if you go ahead and paste the same variable here, I think it's going to work. It's going to show you some insights about it. So it's just a copy data set that you're going to get. So no pretty big deal there, but yeah, when you think of training and the testing, so you need to segregate them as values. So CP time is basically where you're actually giving the dose time, and all these things come out in the picture. So when you are thinking of 32.62%, there are doses in which 24, 48, and 72 instances were taken in a simultaneous fashion. So this is where that patch is being analyzed here. So when you go back and you take an histogram study, histogram is basically how many times it's being repeated. So 0, 1, 2, 3, 4, all these are just the resources which are getting. So in 64 is like optimized version of your 64-bit of int. So it's being optimized directly from the memory. I think I've given the link to the code there. So guys can go to this thing here, so you can start working on uh, this data set completely. Yes, I think let's just go back to our plots which we are talking about. So yeah, CP time for 24 is basically taken here and considered. So a lot of instances, man. When you start thinking about all these, right? Every time when you are thinking of CP of 24, 48, and CP time of 24 without the control and all these things, it's going to be an histographical study where you are going to analyze what should be my time of analysis at every dosage that I'm giving. So it's going to help you with all these. So that's why I told you this is for data visualization. Data analytics is going to be the next part of it. So it's going to be a part 2 and we'll be coming out with it very short time. So stay tuned, folks. We'll be coming out with its part 2. Of course, we'll be getting the 50.96 for the D1, and the dose 2 will be getting 49.04. Yes, we have an accurate result here, much accurate than what Kaggle showed there. So yeah, I'm happy about it at least. So we have got some results. Fine. So yep, it's an histogram of 0 to 0.6 instances. So fixed size I have kept it as 15 as the width and 25 as the height. So it's here, so much greater, and it's amazing. All these plots. Orange ones are actually for the 24. So those one I have kept the blue. For the dose one, you can have a controlled entity where you can think of the his type as a bar and you can start working on it. So next description will be the count of dose one, dose two, and everything. So it's being given here. So mean of it, you need not have to write code for all this. Pandas has much of so much of support in which you can start writing something kind of as and describe and everything. Okay, I think I should show you this thing, I think so. I think probably I'll be going to take one data frame here. I think train feature set. Yep, I'll be taking train feature set and I'll be taking the info of it. Wait, I'll show you that info and it starts working. So it's like 31.1 MB. So of course data sets are like amazing, man. Train feature sets actually comes out to be 149.1 MB. So now on memory, it's been optimized like 32.11 MB. Such humongous data set is being optimized with one simple set of functions which we have written through NumPy, obviously. So I think probably NumPy is going to give you a good support in the future. So make sure that you guys are seeing the NumPy version of everything that you are seeing. So, yep, you know these are all the plots which come on with. I'll explain you these plots. Standard G values are basically the histogram of how much of doses and what are the things which are going on. So all these things are coming out to be picture, and 25, 50, 75 are the maximum values which are going to see, and all these max values is 2.33. So 2.33 has been not plotted here. What is it? Okay, sorry for that. It's like most of it is like 1.33. So 1.33 also has been plotted 1.5, and all these q values which are being found in 1, and this ratio has been plotted here. So yeah, it's pretty cool that it has taken out. So 2.33 and max values have been plotted here. Like 2.5 is the max value and 2.33, yep, it's been plotted here. It's good. And let me just run it here. So yeah, so seeing all these results, like when it comes in the float format, it will be optimized for something. All these blocks plots are basically one such entities where you're thinking about what to be added in the real time. So when you're thinking of subplots, you are going to take some standard values of g variables and the c variables. Of course, I can say what is g variables and the c variables. G variables are those techniques where you are just going to take every single values and just going to say whether this is optimized for human trial or not. C variables are going to be the dosage values which you are going to take, and everything is going to be done. Oh my god, I think I forgot to run all. I think I'll run all. I think better we just run all because it will be more feasible for me in order to get that. So I forgot to run some cells, right? So obviously it will be showing an error. Oh, decrease to 15. Sorry folks, you might be missing my talk, but yeah, I'm available 24 bar 7 bar 365. So yeah, you can. Oh, it's been in GPU. Fine, it's been in GPU. Of course, we got the plots. Of course. So now let's just plot come. Of course, there is standard deviation in the dose 1 and the dose 2. So standard variables with the control has been shown here. So make sure that you're running everything in control. So all these things were without the control what we shown. All these things above are with controls, and these are without control where memory is not being completely like uh, utilized, and also trials which have been controlled also have been considered here. So without the control, yep, we have got similar and accurate result. So now all these results what we have got is like favorable in order to do an human trial. So this is what it says. Of course this notebook is very vast because when I write single line of code and I start executing it, it was not coming out to be proper, so I had to do something. So I had to write everything in a cell. So sorry folks if you are like, what is this, it's so complex. I can't help it, it has to be complex. So standard variation of g duration is 72 and it's 48 here, so we have to just start working on it. So yeah, these are the scree plots where you are taking, these are the confusion matrix which you have plotted, and PCA comes up. PCA, we are going to get the same results. So these are the reverse elbow curves which you are going to see. and these are the trials which can be going through, and these are the values which can be variants of the G in terms of PCA has been considered. So every time when PCA has been applied and all these data sets have been featured and everything, so I have written some functions for it, so I'll be getting some plots here. So yeah, I've got the confusion matrix. So if in future if you are writing your own paper, you can start working on these data sets. So that's all folks, I think. Thank you so much for your amazing time. I'll be linking the notebook and the I've been linking the notebook and the slides in the description, so stay tuned for it. So yeah, bye folks.